Hello, everyone. Thank you for your interest in the Biostatistics and Modeling Core at the San Diego Center for AIDS Research. My name is Dr. Tommy Gaines, and I'm a leadership trainee in the Biostatistics and Modeling Core, which we also like to refer to by its acronym, the BAM Core. Thanks for joining us to discuss our new and exciting quantitative working group. Many of our local CFAR members have used the BAM Core services to provide quantitative support on their research projects. But as an overview, the BAM Core exists to provide expertise, collaboration, and training in the areas of biostatistics and infectious disease modeling. The BAM Core is led by our co-directors, Dr. Sonia Jane, Dr. Natasha Martin, and Dr. Victor DeGratola, and then with myself, Dr. Tommy Gaines, as a leadership trainee. Now, we're welcome today to have Dr. DeGratola join us to introduce this new and exciting working group. Dr. DeGratola is a professor of biostatistics, and his research is focused on the development of statistical methods to respond to the HIV epidemic. Under his leadership, our quantitative working group was launched in the fall of 2018. So with that, I'd like to welcome you, Dr. DeGratola, and start off by just asking you to provide us with an overview of what people can expect out of the quantitative working group.
The quantitative working group is a place where clinical or laboratory scientists can get input and engage with quantitative scientists on developing the best approaches to deal with some of the challenges that we face in trying to control AIDS epidemic. We've had a number of seminars that have addressed the increasing use of molecular surveillance in HIV policy development in order to try to find the best way to target field resources to control HIV. These approaches involve getting HIV genetic sequences from individuals at the time HIV is detected and making use of these sequences to try and make inference about transmission patterns throughout a community, and thereby identify the best way to locate people who might most benefit either from taking PrEP if they're uninfected or by receiving immediate ART if they are infected. Another area in which we've focused has been on the control of HIV within hosts. The ultimate goal is to eradicate or at least reduce HIV reservoirs. And some of the issues that have arisen are how we can make use of single-cell modeling derived from information obtained in single-cell HIV, single-cell RNA-seq experiments in order to make inferences about the genetic processes within immune cells that lead either to replication of HIV or in some cases to the cell becoming latent and therefore evading the impact of HIV treatment.
Wow, so pretty much two areas, broad areas that are being covered within the working group, controlling the epidemic as well as looking at controlling HIV within the host. And since we have these two broad areas, if you could speak to some of the challenges that have been common across the project so far presented to the working group.
Yes, well, it is interesting that although we're working in very different areas, largely guided by the investigators involved with the CFAR, there are some challenges that cut across all of the projects. One is increasing high dimensionality of data, and that just results from the fact that there's so much more that we can measure that was just science fiction when I was starting my career 30 to 40 years ago. Of course, we can do HIV sequencing from HIV that is obtained through blood or from other tissues. But we can also look at the genetic processes within individual cells through RNA-seq experiments. And this leads to a huge increase in the dimensionality of the data. It's also the case that because our CFAR investigators are working with public health departments or have large projects on their own, and these public health departments include not only San Diego, but New York City, Los Angeles, and Mexico City, we also have a huge number of people from whom we obtain information. And the question is, how do we analyze data where we have such high dimensionality? But it's not only that that we're obtaining a lot of information, like HIV sequences, not only of this virus, but also of the human DNA. We also have very complex correlation structures among these data. For example, if we're taking sequences of recently or recently diagnosed, we want to understand how these sequences are related phylogenetically and what are the implications of that linkage to or inference on transmission pattern of HIV. So this leads to very complex correlation structures. And what we need to add to our biostatistical and modeling armamentarium is a deeper understanding of network analysis. And network representation has the advantage that it allows not only for complex correlation structures, but for structures that evolve over time by taking dynamic networks. For example, within sexual networks, in which people may change partners over time. But this is also true of networks of genes within cells that undergo changes as the cell moves closer to mitosis, for example. And so dynamic network structures are increasingly important for the investigations that we undertake. The other issue is that there's very complex sampling. We can never sample all of the people within a sexual network, even if we have a large area of coverage, because there are people that have not yet been tested or people who have been tested and may even have had their virus sequenced, but the sequence is not in our database. And when it comes to the laboratory experimentation, the RNA-seq, we can have genes that, though they are expressed at some points in the cell life cycle, are not expressed at the moment that the sequence is done. So we need to take into account the fact that our representation are imperfect of the mechanisms and the epidemic processes that we're studying. There are no perfect fixes for any of these problems, but there are ways to make progress using recent and ongoing methodologic developments. And that's an area that I think makes our quantitative working group a very energetic and exciting place to participate. The goal here is really to see how quantitative scientists can collaborate most effectively with clinical, laboratory, and public health scientists in order to get control of HIV, both within hosts and across individuals in a population.
Yeah, this is quite innovative. And I think to let our audience members know who they can expect to be around them, not just quantitative experts, but who else are typical attendees.
We've had clinical scientists who are developing, for example, grants for NIH or other funders who contribute, and we've had laboratory scientists. We've had quantitative scientists covering the range of mathematical modeling. classic biostatistics, and networks.
Great. So it's not a group just to calculate sample size for people.
No, although we all end up doing that at some point. Thank you very much, Tommy, for allowing me to speak.
Well, thank you again. And for those who are interested in attending one of our quantitative working groups, we do host monthly meetings. Please reach out to Bryna Block using the following email address. And with that, I'd like to thank you again for your time, acknowledge our funders, as well as the opinions that were expressed during this brief moment are those of Dr. DeGrotola and myself, and not the university. Thank you.